X-ray beam. Why do you use X-ray beam, by the way? Why do you use a beam? Why do you use a X-ray beam? So how does this crystallography work in the first place? So tell me, what information would you get? Any idea?
I mean, they're probably using the X-ray because it's got the smaller wavelength.
Okay, good.
And for some reason, that's useful for visualizing, but I don't know why. Or
Okay.
I don't know more than that.
Okay, so that's a good point. But now, where do we get the X-ray beams? So there's only one place you can do it, in Canada at least. In Saskatoon.
What is it, like a hadron collider? I have
know?
no idea.
So you should Google in Saskatoon, say X-ray crystal structure extraction, or extraction of crystallization data, or whatever it is you can call them. So have you heard about synchrotron in Saskatoon?
Is that the particle accelerator?
Uh-huh.
Is that what it's called?
That's called, it's particle accelerator. Yeah, exactly. Okay, see, we don't know anything about it, all right? So we all sitting in the same, we all are in Saskatoon, and this is the only place in Canada where we can do this work. So anybody who's doing it, you see where you are, actually a pretty good place, actually, a pretty good place. So whoever is studying X-ray in Alberta, Toronto, or British Columbia, or Montreal, or whatever, okay, they all have to come here. Okay, so this is in Saskatoon. Okay, so this is a very, very high energy machine. Okay, and it generates such a high speed accelerator, a beam that can penetrate through the crystals, and it diffracts, and each diffraction emits the electrons, and then you synthesize those information into structures, like what are positive, what are negative charge molecules, and put them into different models and study the structural information. You get it? Okay, so and this is why precisely one of the goal of SGC, so SGC is it will give you different information. Okay, okay, so it's called Structural Genomics Consortium. Okay, so that's one of my former colleague, Al Everett, in Toronto. So basically, it's a very big a resource center. So basically, you can have basically the goal of the Structural Genomics Consortium is to basically crystallize every single human protein and then deposit. So you can just so these people have got so much money. They are so partnering with a lot of industrial people, and basically they want to study the structure of these proteins and how drug development initiative. So why you think studying structures are very important for drug development? Why? Tell me why. So why pharmaceutical companies are very much interested in investing on the drug discovery?
So you can find what spots on the protein might be good to target with drugs.
So how do you do that?
Like if you can figure out where the active site is, you can kind of see
That's
what
right.
drugs might inactivate it.
That's right. So active site. Okay. So precisely is a good point. So studying an active site where the drug binds. Okay, so drug binding to an active site, okay, on a protein is very, very important for pharmaceutical people, because then you can modify, or then you can actually target those active sites, and then you can engineer whatever you want to do. Okay, so and that's one way to do that is actually called chemical soaking or crystal soaking. So what does it mean? Crystal soaking means basically you take the crystals and you soak them into the drug molecule, whatever the drugs are. Okay, so and now you're going to have the drug binding to an active site. So it will allow you the crystals, the chemical soaking or the crystal soaking experiment would allow you to precisely pinpoint where are the active site of the drug on a protein. Okay, so and that's what it's very, very powerful method. Okay, so same thing like the way these guys are showing here, the tertiary structure of this myoglobulin. So basically, imagine this is a drug molecule instead of a heme group, and you're just basically finding where the active sites are. Okay, but this is actually showing where the heme group is bound to the myoglobulin. Okay, and this is one way of looking at it is through a contour, surface contour image, and this is a ribbon-like structures. Okay, so basically showing where the iron heme group is actually are located in the protein structure. Okay. All right. So now here is one another secondary structure example would be, let's say, fibrous proteins. Okay, so basically the alpha helix. So we talked about alpha helix, we talked about beta conformation. So alpha helix, keratin, feathers, nails, all those things are actually alpha helical. These are usually typically cross-linked by disulfide bonds. Okay, and then the beta conformations are fibrin. Okay, and there's also collagen is there. Is another example of fibrous proteins with the secondary structures. We will talk about each one of this example down the road. Okay, so but so this is just an example to show what are different types of fibrous proteins, and whatever secondary structures they have. Okay. All right, so here's one example. Is the alpha keratin. So here's the alpha keratin is the alpha helix here. That's an example we're talking about right now here. I guess it's basically so this keratin is nothing but we all have a keratin, that's air, So it's basically it's an elongated helix with somewhat thicker elements, near the amino and carboxy termina. Okay, so. And usually, when these structures are combined, in a higher order structures, you can call them as a protofibrils. Okay, so basically, so all these elements, so the protofibrils, nothing but all the protofilaments are bundled up together. Okay, in higher order structures, and those are called protofibrils. Okay, so basically the alpha helical structures, those are arranged, in a higher order structures are called protofilaments or protofibrils. So there are about 4 protofibrils. The 32 strands of keratin are there, all combined to form an intermediate filament. Okay, so and these chain coils or the substructures they all are interwound together. Okay, and this is an example of an alpha keratin. Okay, so where you can see the protofibrils, how they all are combined together into a higher order structures to form an intermediate filament. And this is a cross section of an hair here. This is where you can see the protofibrils. The protofilament, you see how they are actually bundled up nicely here. And these are intermediate filaments here, okay? And you have alpha helix here. So, okay, so, and then you have a structure of a collagen here. So structure of a collagen is one example of the fibrous proteins, okay? So here's one example, okay? So collagen, we talked about alpha keratin. We're talking about collagen right now. Okay, so collagen, is an important constituent of the connective tissues. For example, they are present in tendons, cartilage, bones, cornea. Okay, usually each collagen that's got is so these collagens are left-handed helix. So we talked about left-handed helix and right-handed helix. So which ones are right-handed helix?
Alpha helices.
That's right. So here you have this collagen is a left-handed helix. Okay, so there are 3 collagen chains are there. They are intertwined, on a right-handed superhelical triple helix. And again, the triple helix are having a very high tensile strength. And these triple helices are all assembled together to form a collagen fiber. For example, here. Okay. This is the structure of the collagen fibrils. So the collagens are a rod-shaped molecule. They are very rod-shaped molecule. And you can see that they are all very intertwined. Okay. So the collagen molecules, fibrils are made up of collagen molecules. Okay, and these are aligned in a staggered fashion, and they are cross-linked. Okay, the cross-linking is for providing strength. Okay, so now the cross-linking here is different than the cross-linking I'm going to talk about right now. Okay, so whenever you have a so what are cross-linkers, by the way? Let's talk about cross-linkers. What are cross-linkers? Why do we use cross-linkers? Or where do we use cross-linkers?
You can cross-link to maintain structures when you're going to put it through something that's going to be stressful.
Okay, so, but they are good or bad?
What's that?
Are they good or bad?
Depends on the purpose that you need it for. If you're trying to make a rigid structure, then you need cross-linking to have more bonds and more rigidity, but it depends, I guess.
So, so the cross-linkers you can use, for example, okay, so let's say in protein isolation, let's say, okay, so if you want to study a membrane protein, or those membrane proteins are very highly abundant in the cell. So our membrane proteins are very highly expressed. Okay, let's take let's divide this into various groups. Okay, so let's take keratin. Let's take GPCRs. Let's take chaperones. Okay, which proteins are very high abundant? When I say highly abundant, what does it mean, highly abundant?
Just means there's a lot of them relative to other things.
So is keratin protein is highly abundant or low abundant protein?
It's really abundant in skin cells and hair cells, but depends on what cell you're talking about.
Well, I'm just talking about is keratin in general. How are they expressed? Very high or very low in the cell or in any cell? Are they highly expressed?
Overall, probably pretty low.
Pretty low?
I would guess so.
So keratins are very highly expressed, okay, are very highly abundant protein, okay? What about GPCRs, which are membrane proteins? Are they highly abundant in the cell? So I think here you got to keep in mind the context here, okay? Highly abundant and low abundant. If you take any physiological sample, just any normal sample, there are 20,000 proteins out there in a human body, am I right? You think all of them are expressed at a very high level in the cell? No. So what are the most highly expressed ones? I talked about keratin, we talked about. What about chaperones? Are they highly expressed? Are they?
Yeah, the chaperones are highly expressed.
Why, Brian?
because they're involved in forming more proteins.
Yeah, exactly. So they are very highly expressed. But what about GPCRs? Are they? Are receptors? Are they?
Yeah.
Usually, yes.
We say the receptors were one of the most prominent classes of genes.
Yeah, but that okay. So I mean, I guess you guys are getting confused because of the prominence, because of the important role. Doesn't mean they should be highly expressed. You know what I mean?
So we're differentiating prominence and abundance here.
They are different.
Okay. Okay. Yeah. I see.
Yeah, they are different. Okay. Just because you have a protein with a critical function doesn't mean those are highly expressed in the cell. Okay. But they are critical. It doesn't necessarily mean they're highly expressed in the cell. So the receptors or the membrane proteins are the ones that are very expressed at a low level. Okay. When I say low level means, is it very hard for you to isolate a membrane protein? Is it very easy? It's not. It's very, very hard because you have a lot of lipids, am I right? So a lot of alpha helices. So the more the alpha helices and the beta barrels are there, it's going to be more difficult for you to isolate the membrane protein in the first place. But I mean, that's isolation, am I right? But how are they even expressed in the cell? These proteins are very critical, but it's okay. Let me put this way, okay? Receptors are very important for many functions, like signaling functions, am I right? Yeah? Yes or no? So receptors are very important for signaling, cellular signaling. Is that right or no? Yeah? Okay, so, and imagine glucose. We need receptors that bind to the any agonist to amplify it. Yeah, is that correct? To produce insulin, to produce, synthesizing the glucose. But what happens if the protein itself is expressed at a high level than the physiological state? What happens? So do you think like having, so if a low abundant receptor is allowing to amplify a glucose molecule from 1 molecule to 100,000 molecules, based on its physiological condition, what happens if that receptor is highly expressed in the cell? That means instead of 100,000 molecules of glucose, there's going to be 1 billion molecules of glucose. Yeah. Do you think it's going to be helpful for the body? No. It's going to, everybody's going to go into diabetes, okay, pretty much, okay? Yeah. So the point is, that's what I'm saying. The prominence and the expression are two different things. Yeah. So, which means even a protein, if they are expressed low, doesn't mean it undermines their critical function. Yeah. Because these are very important molecules, and maybe there's a reason why they are not expressed like chaperones or like keratins. You know what I mean? So studying those type of proteins are very, very what? See, there you go. My Alexa is talking here. Anyway, so I don't understand where it's coming from. Okay, all right. Hold on. Just hang on a minute. Hang on a minute. This is ridiculous. Okay. So the question here is, receptors are very, very important. There's some receptors, like GPCRs, they are expressed at very, very low level. Okay, and the cross-linkers are absolutely critical because, for example, if it's very difficult for you to isolate a protein, from the membrane, and these cross-linkers can allow to or help to to capture those associations. For example, so let's say you want to study an association between a receptor and a membrane, and a receptor and another membrane protein, or a receptor membrane protein to a non-membrane protein. If you want to study, let's say, this type of cross-linkers, these are amine-based cross-linkers, okay? And these amine-based cross-linkers can help to capture even associations that are not direct, but also indirect, So meaning like, not just because based on proximity, but also to study more difficult proteins, you can use cross-linkers to study protein associations. Do you follow? Okay, so these amine-based cross-linkers are absolutely critical. But the issue here is like, if you add more cross-linkers, then the physiological state would be altered, which means you are going to see associations that are all noise rather than the true associations. Okay, so cross-linkers are absolutely critical for studying protein-protein interactions. Okay, and cross-linkers can allow you to study low abundant protein, or cross-linkers can help you to capture transient interactions. Okay, because many of those receptors undergo interactions that are not easily being identified by a normal biochemical experiment. So cross-linkers can allow or favors to capture transient interactions. Okay, so, and in this context here, in the context of collagen fibrils, okay, so as I said, cross-linkers are very important because they always gives you rigidity or strength. Okay, so, and in this here, the cross-link, the collagen fibrils are made up of collagen molecules, and the strength is being facilitated by these cross-linkers. Okay, so it's very, very important. So this is electron micrographs showing this rod-shaped molecule of the collagen fibrils. Okay, all right. So now one other example, we will talk about it, and then we will, maybe we can just wind up here. 35 slides. Okay, so silk fibroin is another example where fibroin is the main protein in the silk from moths and spiders. So they do have an anti-parallel beta sheet structures, and alanine and glycine are the ones that are very important for the beta sheets. And the structure is very much stabilized by the hydrogen bonding. Okay, so and here is the structure of the silk. And this is the protein fibroin, actually, how they are arranged, actually. So they have an anti-parallel, you can see there are anti-parallel beta sheets, and they are rich in alanine and glycine residues. Okay, and they are very closely packed. Okay, and this is an example of a ball and stick view of seeing the structure of the silk or the fibroin. Okay. So spider silk is another example where you can see, usually, and these are, this spider silk is also another example of a fibroin. These are fibroin-rich material, and these are stretchy material, and usually they are very strong. Okay, and so these are different examples of fibrous proteins. Okay, fibrous proteins, and we talked about all these fibrous proteins here. So we talked about keratin, we talked about silk fibroin, we talked about collagen. These are different examples here. Okay, so I think for the midterm, we can stop here. Okay, so we can stop here. Then from next week onwards, well, not next week, a week after, we can talk about this motifs and everything. Okay, so we'll stop here for the midterm here. Okay, so now what I suggest to you guys is take a break for I don't know, for 15 minutes, let's say. So we'll start at 7:25, and then what I want you guys to do is actually let's go over the materials, come up with like about 30 questions, and then we will then we will use those material to come up with some decent questions for the midterm. Okay. So let's reconvene here at 7:25, guys.